receptor-mediated endocytosis and signaling endosomes, those both could be covered in a endocytosis section or in a signaling section. And the same is true of the next topic, really the last thing that's going to be part of this module that we're going to cover, and that is exosomes. To get there, we need to deal with this issue of multivesicular bodies that we've been talking about, MVBs, for quite a while. So let's talk about how they form and cover these escort proteins that were mentioned in the microautophagy section. So now there is a variation on the endocytic pathway that begins in some cases at the plasma membrane, but in this diagram it begins at the early endosome. It's going to determine a different fate for this particular receptor-cargo combination. And it begins with the involvement of a single ubiquitin moiety added to the receptor. So this is an example of monoubiquitinylation that I alluded to way back when we were talking about the proteasome. This ubiquitin does not signal the involvement of the proteasome. Instead, it dictates the way in which this membrane is going to behave in subsequent steps. So what's shown in this pathway is the maturation of the endosome involving that inward budding process that was introduced by the microautophagy mechanism. But what's going on here is the formation of membrane-enclosed luminal material that contains vesicles, so-called intraluminal vesicles, that have cytoplasmic components inside them. So these are inverted relative to the regular endocytic vesicles that we were making by budding off the plasma membrane, because those things have the extracellular space, or the lumen, that would be the gray area, inside. These have the white cytoplasmic area inside. So that reaction is not one that we've talked about at all. So we need to account for that. But for now, just realize that ubiquitin is one of the main signals that says, rather than just fuse with the endosome, rather attach to it and then undergo that inward budding to produce these intraluminal vesicles. And in the process of going all the way through to the endolysosome, the late stages of endocytosis involve relatively large structures that have a whole bunch of these intraluminal vesicles, and that's why they're referred to as multivesicular bodies. So the early stages in the process of that inward budding, so here we go, here's the lumen and the cytosol. So this is the cytoplasm being incorporated into the interior of these vesicles, which are essentially inverted from relative to the cargo vesicles. We have another phosphoinositol, in this case is PI3P, that is one of the specific signals in that ubiquitin, monoubiquitination. So we don't have 4 or 8 or whatever, we have one ubiquitin residue, and that's that. You can have multiple monoubiquitinations, but only one serves as a recognition site for the binding of a protein called escort 0, and that triggers sort of a recruitment of additional escort factors that lead to something that looks like this. So complexes of proteins that make specific contacts at regions of the endosome membrane on the cytoplasmic side, and once they form, they essentially move together in this direction, so that pinches off the membrane into the interior, into the lumen of the endosome, and it eventually fuses to produce these interluminal vesicles. So now, probably the most common consequence of that is the maturation of this into the late endosome and then fusion with the lysosome. So if you can imagine dumping all those hydrolytic enzymes into here, then everything gets destroyed. This membrane, the cytoplasmic components, and everything that's sticking out gets destroyed. So this is a very nice way of getting rid of all of the entirety of the structure of a receptor, because the other possibility would be that that receptor is right here as a result of a fusion reaction with the extracellular part from the membrane now facing into the lumen. So it would be organized like so, and you could imagine that, well, we're not actually degrading that part of the receptor using that mechanism. But using these interluminal vesicles, the whole thing gets destroyed. So that's one advantage of that mechanism. But I don't think that's why it exists. I think the reason it exists is another possibility altogether. And that is, imagine if you take that multivesicular body and bring it to the surface of the cell, bring it to the cytoplasmic surface of the plasma membrane. Then if you fuse it with the plasma membrane, then this entire thing gets released into the extracellular space. So here's a diagram that shows what I'm talking about, although unfortunately for some reason they completely reversed their color scheme. It drives me crazy. So now they're making the cytoplasmic environment gray and the luminal surface white, so don't get too confused with that. But the fusion of a multivesicular body with the plasma membrane would essentially release this vesicle, because this is the membrane that fuses, so it's released more or less intact into the extracellular space. So another thing that they use this diagram to describe how viruses pick up an envelope and then are released. So the viral genome for something like a coronavirus would be in here, and the membrane that it picks up was stolen from somewhere in the trafficking process. So that's one application of this idea. But I want to talk about something that the cell does, whether, that's not necessarily infected by a virus, that releases essentially packets of signaling material that can be, in some cases, responded to by cells that are a long way away from the cell that released those things. And those signaling packets are referred to as exosomes. So here is a diagram that's really nice. It's from a recent review of the importance of exosomes, in particular in the so-called EMT, epithelial mesenchymal transition, or mesenchymal transition. Very important oncogenic process by which epithelial cells become migratory. It's one of the fundamentals of metastasis, for example. But it's a very nice description of endosomes in general and how they might function even in the absence of cancer as signaling processes. So the thing about this diagram is it begins over here, which is a little bit weird, but this is what we've been talking about. Early endosome formation as a result of early endocytosis, the production of MVBs, which are called MVEs in this diagram. Multivesicular endosomes is exactly the same thing. And this is the signaling packet that I'm talking about. So when it fuses, when the MVE or MVB fuses with the plasma membrane, these things are released into the cytoplasm. So if you imagine that there is something here that triggers a developmental change, something like a growth factor, some sort of developmental signal, which is going to be true. There is Wnt in some of these, if you know Wnt from developmental biology or cancer. Something like that, then this exosome can then turn around and bind to the cell that released it and trigger a developmental change in that cell as a result of the release of the signal into the cytoplasm of that cell. So because this cell is signaling to itself, that's something that we refer to as the autocrine system. But because this is in the extracellular space, this exosome can travel. If it travels to the cell next door, then it might trigger a developmental change in that cell, either through fusion with the plasma membrane, releasing the signal inside, by binding with to a receptor on the plasma membrane, or by endocytosis. Any of those things is possible. And because that's one cell signaling to a neighbor, we would refer to that as paracrine. So while we're on this diagram, there's an additional component that I haven't mentioned. We talked about the possibility that there's some sort of proteinaceous signal in there, but experimental evidence has proven time and time again that these exosomes tend to be enriched in small RNA molecules, in particular small RNA molecules that are involved in signaling, the so-called microRNAs that are abbreviated like so, or very often given the mir prefix and then some number to indicate which one we're talking about. And some of these have become very famous. I'm not going to talk about this phenomenon. Once again, it's gene regulation, and we don't do that. But the fact that those microRNAs are found very commonly in these exosomes is a pretty good indication that this is a way for this cell to signal to this cell and actually control gene expression, in some cases of some very important genes in that cell. So you've got microRNAs. In some cases, those exosomes have been shown to contain proteins of the class referred to as integrins, and we haven't talked about those yet, but we will. So just save that concept for later. Integrins, that's an R. And they also contain, as I mentioned, Wnt is a fairly common constituent of these exosomes. There is also ubiquitinating enzymes that are important in controlling regulatory pathways, particularly in the case of those cancer-associated exosomes. So that sort of collection of material is shown down here, microRNAs being probably the most common, and along with proteins. In some reports, there's DNA, in some cases intact messenger RNA. A variety of integral membrane proteins are present. Some might be involved in the formation of exosomes. Some might be involved in the specific recruitment of microRNAs, so that it's just not a random collection of microRNAs. It's actually specific ones that are designed to give very precise instructions to this cell. And although this arrow indicates that this cell is right next to this cell, there's nothing to keep this concept from extending to tissues that are a long way away, and that this method of signaling, as long as this exosome is relatively stable, you can have one tissue communicate with another tissue at a great distance in the position in the multicellular organism. So that would be an example of an endocrine type regulatory, basically a hormonal almost signaling mechanism. And that's really what concerns oncologists, the ability of this cell essentially to convert tissues in another location to become cancerous. And of course, metastasis is why so many people die of cancer and why so many cancers can't be treated. So I guess if there's a bright side to this mechanism, it is that these exosomes, by virtue of being extracellular material, they can be collected relatively easily, and all of the components can be detected using modern methods of detection, antibodies, and RT-PCR, et cetera, so that it's thought that these exosomes might be might contain very good biomarkers that might provide an early indication of a cancer that is about to spread, but that it would be detected before it spreads and therefore could be treated with anti-cancer drugs that work pre-metastasis. Okay, so I'm going to stop here, and this is actually really the end of this module, and we'll move into the specifics of signaling reactions with the next video.